All right. Okay. So I see your slides are up. So let's go ahead and begin. We are just 1 minute after the hour, so let's go ahead and start. So it's my pleasure today to introduce Dr. Justine Hansen. She received her bachelor's degree in neuroscience in 2020 from McGill and her PhD with Bratislav Misic's lab at the MNI. And during her PhD studies, she published so many papers and tools that lots of us use in the field, including my lab, I will say. We love her tools. Her careful and comprehensive multimodal approach to understanding the brain with a focus on individual variability is something that I greatly admire about her work. And each time I see a new paper with her as first author, I know it's going to be a good one, and I'm excited to read it. Others clearly think this as well. She's received the Brain Star Award from the Canadian Association for Neuroscience in 2022 and the Carl Zillis Integrative Neuroscience from OHBM in 2023. And she just started a postdoc fellowship at the Children's Hospital of Philadelphia at University of Pennsylvania, where she works in the Brain Genes Development Lab with Dr. Aaron Alexander Block. And she'll be presenting today her work, her recent work on understanding brain structure and function by studying the brain as an integrated whole. She will discuss how local biology can mirror global dynamics and wiring, and how individual differences can inform our interpretation of spatial relationships between brain features, and how brain structure reflects the body's biology and health. And I'm very excited to hear her talk and can't wait to hear about her newest work. So please take it away.
Thank you, Amy. What a nice and generous introduction. I'm very excited to be here to show you guys my work. I like to start with this sort of thought experiment where I give you a brain map that represents some sort of feature, some sort of biological feature that has been measured across the surface of the cortex. And I just ask you, like, what do you think this is? And maybe you're a cognitive neuroscientist, and then you say something like working memory brain activation. That could be the case. But if I tweak this scenario a little bit and say that you're some other neuroscientist with some other domain expertise, you could have said like a whole host of different biological features across a whole hierarchy of spatial scales. And you probably would have been pretty like right all the time. And that's of course because the brain is an integrated organ. And so all of these different biological features are working simultaneously, and somehow the brain is integrating all of these to give us, my ability to give you this talk right now. And that kind of leads me to the main premise of a lot of my work, especially during my PhD, which is that to understand brain organization and function, we really need to explicitly consider the shared influence of all these different biological determinants rather than treating each biological feature as its own isolated perspective. Now, the good news for me and for us is that multi-scale biological features are measurable, and they're available through all these different like technologies and techniques like molecular assays or histology or in vivo imaging, even meta-analyses. We've gotten a lot out of that. And so it makes it more and more possible to try to dig into the different biological mechanisms that are at play throughout the brain. Now, of course, like the challenge with this is that no one person can be an expert in everything. So to kind of go down this route, we are opening ourselves up to the vulnerability of not entirely knowing what we're talking about, because we can't know everything about everything when we start to bridge different data sets and different spatial scales and different techniques. On the flip side, Brad Wojtek put it really nicely. He said, "We are data rich but theory poor." And that's because we have all these different, like, sort of semi-siloed labs that have been collecting so much data and are sitting on all this data. And it's not necessarily closed, it's all there, but we're not necessarily making connections with one another. And as a result, we're not coming up with theories that have to do with the brain as a more integrated whole rather than a very specific facet of brain organization or structure. So that is where I come in and try to ask these questions about things like how are different levels of brain organization conceptually related? How are they quantitatively related? How do I try to bridge these different gaps, but also acknowledge my own limitations and my own knowledge, given that I can't be an expert in everything and I don't know the nitty-gritty of every single data set that I've worked with, although I would love to know the nitty-gritty of every single data set that I've worked with. So I'm going to start with this chapter about mapping molecular phenotypes to whole brain connectivity and dynamics. And I like to start with receptors because they're quite intuitive and we sort of know how they operate. There are these proteins, they're embedded within the cell membrane, and a neurotransmitter might bind to it, which is going to change the configuration of this protein and allow for the flow of charged ions in and out of the cell. And that's going to change the excitability of our neuron, which is going to change whether or not it's, firing, and as a result, whether or not it's connecting to other neurons or synapsing onto other neurons. And so we know, like we know, kind of at the core of our, neuroscience knowledge, that these neurotransmitter receptors modulate neuronal firing rate and therefore also its wiring. Now, One of the things I like to do a lot with the work that I do is I take something that we know and then I say, well, do we also know this if we zoom out a lot? Is it the case that your receptor fingerprint will tell you something about the way a brain region or a population of neurons is anatomically connected to other brain regions, or will it tell us something about the population dynamics of that brain region or of those neurons? Given that we know it at the local scale, right? At the same time, I also want to prime you right now. This receptor project, which I will speak very briefly on, that came at the beginning of my PhD. And towards the end of my PhD, I took the exact same logic and I applied it to synapses. It's a little bit less obvious with synapses because it's sort of harder to imagine exactly what is a synapse in the way that we can imagine a receptor is this like protein that's sitting in the cell membrane. A synapse is sort of, well, the morphology can change, the proteins that exist there can change, its location on the cell can change. It's a little bit muddier. And so the logic is maybe harder to follow, but the same applies, right? We know at the scale of the local biology that your synapse phenotype, the way that your synapse is kind of made up, is going to have something to do, is going to have an effect on the voltage dynamics of that cell. So then is it the case that if we zoom out, we see the same kind of global relationships with synapse types? I'll get to that in a second. First, I'm going to start with these receptors. Here is a bunch of brain maps of different receptors. And the point, I guess, is that they are heterogeneously expressed in the brain. And the way I like to think of receptors, I think of them as like ears. They're listening for some sort of signal. And so if you're in some part of the brain and you have some composition of ears and you're listening to a specific signal, you're going to be responding possibly similarly to another brain region that has a similar composition as yourself. So let's see if that's the case. Here's a brain map. I've zoomed into a brain region and I'm looking at the receptor fingerprint of that brain region. So just, the density of, let's say, 20 different receptors there. And I take some other brain region and I look at its receptor fingerprint. And again, the logic here is, this is a brain region and these are the signals that it's possibly listening for. And then I can correlate those two and I get this receptor similarity matrix, which is just a correlation matrix telling me how similar are two regions in terms of the receptors that they are expressing. And then I can compare that to structural connectivity coming from diffusion-weighted MRI. This is just an adjacency matrix telling me whether or not two brain regions are physically connected or anatomically connected. And what I find is that regions that are more similar in terms of their receptor fingerprints, so in terms of the receptors that they're expressing at this brain region, they're more likely to be anatomically connected than not connected. And likewise, if we extend that to functional connectivity, this is coming from fMRI, so just telling me how similar are two brain regions in terms of their functional time courses. It turns out that regions with similar receptor fingerprints tend to be doing functionally similar things. So in this case, what I mean is within functional networks rather than between functional networks. Okay, now let's get back to the synapses because that's a little bit more new, and so I'm kind of more excited. I'm a little bit over the receptors, I'm not gonna lie. Um, Here's a brain map. This is the mouse brain. So the Synapse project was done in the mouse, and this is my first time going to the mouse, so I didn't really know what the mouse brain looked like. So this is like honestly for my own sake, so that I know where the regions are. And what you need to know is here are a few different brain maps for different synapse types. And what I learned coming into this project, I came into it thinking, okay, synapses, there's excitatory, there's inhibitory, there's broadly modulatory. It turns out that even within the excitatory subdomain, these are all excitatory. There are all these different types of synapse types or synapse phenotypes, and it comes down to their morphology, it comes down to the proteins that they're expressing, and there's all this electrophysiological work showing that, especially from Seth Grant's lab at the University of Edinburgh, showing that the voltage dynamics of the cell is dependent on the synapse that is located there. So we have the same logic as the receptors. Given if you have a brain region and you're expressing certain types of synapses there, what are our dynamics going to look like, and what's our structural connectivity going to look like, given that you have this unique dynamic profile? I'm not going to go into much detail about why I'm calling these synapses. It has to do with the protein that they're expressing, but definitely feel free to ask. I'm happy to go into more detail there. Okay, so the main question that's driving this project is the regional synaptic composition related to regional activity? So again, we know this is the case at the microscale. We know this is the case at the level of a single cell. Now, can I zoom out and see that it's the case across the whole brain? And maybe more specifically, in this project, unlike the receptor project, I had like 20 receptors. So I could do this receptor similarity matrix and ask how similar are two brain regions in terms of the receptors that they express. In this project, I really have two, kind of three, but I'm not really going to talk about this one. So let's just think of it as two different synapse types. So I'm not doing analyses where I'm looking at how similar regions are in terms of their receptor fingerprint. I just don't have enough synapses for that. Rather, it's given my synapse type, can I associate that with specific features of neural dynamics? So there are a few ways to do that. The first, not very scientific way is to take a brain map of one of your synapse types. So, higher expression up here, lower expression down here, and plot some of the fMRI time series that are coming out of this. So again, this is different data sets, like the synapse density data that's ex vivo and the fMRI, this is in vivo in awake mice. And I'm just looking at time series that come from regions that have low density to high density of this synapse type. And I'm kind of just staring. I'm not even joking. I actually just stared at it and was like, can I convince myself that there's anything, like, do I see anything in these time series? And you can do that for a synapse type and kind of pull some time series, some example time series from low density to high density. And like, what do I see? Do I see anything that is correlated with the presence of the synapse? Now, of course, there's not much you can get out of just like staring at these forever. Oh, the color, I should just say the color of the time series, it just has to do with the ontological structure of the region. So is it coming from the hippocampus? Is it coming from the cortex? Whatnot. So here's the more quantitative way of doing this. I use the highly comparative time series analysis toolbox. This is the one that Ben Fulcher put out when he was, I think, a grad student. And I quantified about 7,000 different statistical time series features on these time series. So things like kurtosis, things like variability, stationarity, how predictable is my signal? If I know the first half of my signal, how well can I predict the second half of my signal? Things like that, how cyclical. And so here are my brain regions, and so each brain region has a time series associated with it. And then here are my 7,000 different time series features. Think of it as like a kind of massive phenotyping of my time series. And I just correlate that with my synapse density. So this is not a line, these are dots. There's just 7,000 of them because each dot represents a time series feature. They're sorted in ascending order. And this is absolute value, right? So I just care about the magnitude right now. And I'm looking at the statistically significant features. And one of my time series, or sorry, one of my synapse types is correlated, this one over here, is correlated with time series features that have to do with the stationarity or the predictability of the signal. And the other one, and this one is much easier to see in the figure, is correlated with the presence of these outlier or extreme events, which we can see these like spikes right here. Now, first, red flag is this motion. You just have to trust me that it's not motion. I did a whole thing. I was like, oh, okay, well, this is probably motion. Do I need to be worried about that? I don't think it's motion. Like there was a whole, anyways, that was like a few months that I went by trying to make sure it was not motion. Um, yeah, so here we kind of see, okay, given the synapses that exist in a brain region, there are potential associations with the types of dynamics that you're showing at the level of like the population, right? No longer at the level of the single cell, although we do also see this at the level of the single cell. Um, and that translates into how connected a brain region is going to be. So here what I'm showing is that one of my synapse types, but not the other, is correlated with how many physical connections you have or your structural hubness, so the anatomical connectivity. This, by the way, is the synapse type that was correlated with the extreme events. I sort of think of it as like an on-off switch. You have these like big action potentials. I mean, it's not an action potential because this is fMRI and it's population dynamics, but you have this big burst of activity. That I think of as kind of establishing or trying to establish these physical connections. And on the flip side, with functional connectivity, I get that my other synapse type is correlated with how functionally integrated you are. This is the one that was predictable in its signal. It was correlated with stationarity and this model, or time series predictability, which just from a purely statistical point of view, we know that the cleaner your signal is, that easier it will be to get correlations, right, in your data. And so we kind of see that reflected in this correlation with functional hubness. Whereas my other one, this kind of structural synapse, doesn't seem to reflect that very much. Okay, so here I sort of hope that I've showed you that both the receptors and the synapses, and really I would argue a lot of the local biology of the brain region, of various brain regions or various neurons, the kind of relationships we see there with structure and dynamics, they translate to the macro scale as well. And so by understanding how the local biology is operating, it sort of helps us interpret the global dynamics and global connectivity at the level of the whole brain. Okay, now what about individual variability? So this is like one of my challenges with the sort of work that I do. A lot of the times, because I'm doing this kind of integrative work that is consolidating different data sets and different data types, and data is coming from different people, and a lot of the times it's group averaged because they can't necessarily share individual data because there are ethical concerns there. And you just can't collect all of this data in, a cohort of people because you would need them to be like both living and dead at the same time, and it just like would not fly. So how do we kind of tackle that? And I don't really have an answer. Like I don't exactly. I think a lot of the work that I do is limited in their questions by this like limitation of individual variability. For example, I can't ask questions about how do differences across people translate in terms of differences in terms of like cognition or behavior, and that's fine. You know what I mean? Like there's science that can address that, and the science I think can't. Science that I do is going to be more about, global topography of brain maps. It's going to be more about what is common across people rather than what sets us apart. But one way that I kind of try to dig at this question of individual variability a little bit is again with this receptor project that, again, like beginning of the PhD, just keeps coming back to haunt me. In 2022, there was this project. We collected data, collected as in asked people if they would share the data they had collected. Um, and we came up with this kind of collection of brain maps related to various receptors and transporters. And I only had the means. I was asking people if they would be willing to share their data, and everybody was very generous, and they shared the group means because they couldn't share the individual data because there were all these ethical restrictions there. And so whenever I got asked, how do these measurements vary across people? I was not able to give much of an answer because I didn't really know. What I did know is a lot of the times I would get multiple brain maps, and so I could correlate them spatially, right? I could say, well, I got this brain map from group A and I got another brain map from group B. They're both supposed to target 5-HT1A. Let's correlate them. Oh, it's very correlated. Okay, well, probably the individual variability is not that high given that these brain maps correlate so well. But that's a slightly different question because that has to do with spatial similarity and not necessarily to do with your actual measurement variability, right? Because you could z-score that and it wouldn't matter. In any case, what I ended up doing is I went back 4 years later and I asked, would you guys be willing to find that data again and then share with me like the standard deviations? Like, obviously, you still can't share the individual data, but can you just give me the standard deviation too? I will just like put a little plug. Like, if you are asking people for data, or if you're sharing your own data, I think we should just like do this from the get-go, like the mean and the standard deviation, rather than just sharing the mean. That was just really dumb. Like I should have done that 4 years ago. But anyways, I kind of went back with my tail between my legs and said, "Hello, I'm back. Can we get that?" And again, people were really generous, which is just so great. I'm a little bit naive when it comes to science because I've had all these really positive experiences of people being like really nice and friendly all the time, and then other people are like, "Really? That's not my experience." And I'm like, "Well, I don't know. That's my." Anyways, okay. So we have these are not actually standard deviations. These are the coefficient of variation because in biological systems, the standard deviation scales with the mean. So this is the standard deviation normalized by the mean. And first of all, it's heterogeneous, not very surprising. And second of all, it is organized in terms of structural and functional regions. Like it's not random, I guess is what I'm saying. And one of the really confusing things about these plots, which I don't really know what to think about, is that a lot of the times the variability is higher in these kind of sensory motor regions, which I wouldn't have expected. I wonder a little bit if this is in part because I'm doing the coefficient of variation, because again, the standard deviation maps are going to look a lot like the means, right? Like they're quite correlated. And here we don't see the sensory motor regions popping up. So there's something about the potentially low density there that kind of makes that variability higher. And it makes me wonder that in like previous or in the work in the literature, a lot of the times people are reporting standard deviations. I wonder a little bit how the story would change if we were talking about coefficients of variations instead, because I think that would be relevant to just know how much things vary given the mean quantity of this measurement in those regions. In any case, here are my coefficient of variations. And, this kind of could be the project by itself. It's sort of just about the information. It's like, well, here it is, and now we know. And, sort of take it as you will. And different people will have different favorite receptors that they'll want to know where, which regions vary more, which regions vary less, or maybe just generally like whether it even varies enough for us to have to care about it, right? But there are different, or this project made me really think about all the different ways we can think about variability. And the one that I'm showing back here with the coefficient of variation, I'm taking a measurement in some brain region and I'm looking at how it varies across people. And that's one way to tackle it. And the way I tackled it before in 2022, when I didn't have access to the coefficient of variation maps, was I was thinking purely about spatial consistency. So I was just saying, well, do the maps look alike? I don't really care about the magnitude of the value. And the logic here, right, is you and I could have the exact same brain map. Like the topography here could be the same, but you could have 2 times as much receptor density as me, and that would end up in a high variability across people, but a low, sorry, high variability here, but high spatial consistency here, right? So low variability in terms of your spatial consistency. And then there's like an argument of, well, which one do we actually care about? Like there's you care about there being different receptor densities across different people if you care about individual differences in biology in the way you might care about individual differences in like your head size, right? And then you care about spatial consistency if you're more just interested in the topography of the map and you're more interested in the organization of the whole cortex. And it turns out there's this other version of variability that actually has nothing to do with inter-individual variability that sort of bridges these two, and that's your measurement variability. And this is just kind of very statistical, but you can sort of imagine if you have very low variability in terms of, let's say, your 5-HT1A density in your brain, let's say they're all ones, then across people, if there's just a little bit of individual variability, that's going to be enough to perturb the rank order of your brain regions in terms of the density of this receptor. And so you're going to get high individual variability if you have low regional variability and a little bit of individual variability. On the flip side, if this brain map is super extreme, you have two poles that are really far away, you have a really high variability across regions in terms of this measurement. If you have just a little bit of individual variability, it's not going to change the rank order of those brain regions. So you're going to be quite spatially consistent. And it might not matter as much that there's individual variability in this feature. So first, here what I'm showing, the orange histograms, this is the distribution of the coefficient of variation of all the different brain regions. So histogram, because we have like 100 brain regions or whatever. And then the dashed line is representing the coefficient of variation of across brain regions. So this is no longer inter-individual, this is now across brain regions. And the argument here is if we just look at the orange histograms, the subcortex looks like it varies more, right? Because the coefficient of variation is pushed to the right. And we can sort of compare across receptors. You know, the serotonergic receptors seem to show pretty low variability. The GABA receptors show higher variability. We can talk forever about whether this variability is biological or methodological, but you can sort of compare them a bit more systematically this way. But then if you look at where the regional variability lies, in the subcortex, almost all of the time, your interregional variability is greater than your interindividual variability. So this dashed line is further to the right than my orange histogram. And what this means is that in the subcortex, I would expect that our spatial distribution of these measurements is much more consistent across people because you vary more across regions than you do across people. But in the cortex, that's less often the case. A lot of the times you're actually varying more across people, even if it's very low variation. Just the fact that it's more across people than it is across regions is already telling me that you're less likely to be spatially consistent. And we can show that this is the case by comparing them a little bit more directly. Here on the x-axis is your mean spatial consistency. So the way I've done this is that in any case where I have multiple group mean brain maps, which is not the case for all the receptors, but for all of these, I'll have multiple different brain maps coming from different groups and I can correlate them. And maybe if I have three, then I can do six different correlations, right? Is that true? No, three different correlations. And take the mean. And so this is just representing how spatially consistent am I. And then on the y-axis, I'm showing what is the ratio of your interregional to interindividual. So in other words, what is the ratio of this dashed line to my histogram? And again, the logic being if my dashed line if my interregional variability is way bigger than my interindividual variability, I'm probably more spatially consistent. And that is what we see with these correlations. This correlation is not statistically significant. And this is not to say that this is supposed to be some sort of like big takeaway result. It's more that when you see the receptors kind of laid out here, you can infer where's the variability coming from. Is it more methodological? Is it about biology? Is it about individual differences? By sort of pinpointing, for example, 5-HT1A, this map it just looks the same no matter how you're looking at it. And so this one, now that I've had several years of kind of staring at this guy I'm super confident is going to look the same all the time. And then there are others where I'm like, yeah, well, this one I think is reflecting more individual differences or methodological kind of difficulties with PET imaging. Okay, so let me bring us back to mapping molecular phenotypes to whole brain connectivity and dynamics. What I've talked about are like really my big takeaway is that we know so much about the local biology. And so if we use that to inform our understanding of global connectivity and global dynamics, I think we get a much better picture, we get a much more comprehensive understanding of where this global connectivity and dynamics is coming from. Now, for my postdoc, so that is the end of the PhD work that I've shared. I'm going to go into my postdoc work, which is like really early. So, you should take everything with a huge grain of salt. It also means that if you have comments and like critiques, it's more likely that I can actually address them, unlike the work that's already published. So you're very welcome to, inform me in the direction that I take my project. What I did in my PhD, I had this like bad habit. Well, I don't know if it's a bad habit, but I had, I guess, maybe a bit of a bad habit of I was always thinking about what am I not thinking about rather than thinking about what I should be thinking about. And in my PhD, one of the things that I was really not thinking about was the fact that the brain is obviously embodied, like it exists in the context of the whole body. And so I started thinking, well, what about all of the influences from the rest of the body? And don't even get me started on like the environment. I haven't gotten there yet. Like, I'm sure I'm sure it's coming. But um, so here's this like sort of a preliminary title, Comprehensive Mapping of Brain-Body Associations Throughout Development. And the inspiration came first in 2013. Ye Tian, who at this time was a postdoc with Andrew Zaleski, she had these two papers that came out. And I'm not going to go into detail about what she did. The point more so was it made me realize the degree to which I was ignoring the rest of the body and the degree to which they are very coupled with one another. And then the same year, this paper comes out. And here I'll go into a little bit more detail on what they're doing. They are taking an MRI of the heart, segmenting the heart, getting all these features of various, like wall thickness and area of various parts of the heart, and correlating that to structure and functional features in the brain, also from MRI. I think they're doing this in the UK Biobank. And they get all these associations, and they're saying, oh, well, heart development and heart structure reflects parts of the brain. And what I took away from this, well, first on the side of Ye Tian's work, we know that brain health and body health are impacted together. We know that they are related to one another. And I don't need to convince you because we feel this every day. If I'm anxious, I will feel it in my gut. Or I don't know, you can feel your heart rate increase, these sorts of things, right? At the same time, we know that the brain and body co-evolve and co-develop. They are not developing or evolving in isolation. And so various growth signals that are being given to, let's say, the heart or just generally the rest of the body, they're also going to be heard by the brain. Now, at this point, it's like, they're heard. Does that mean that the brain is doing anything about it? But I was just sort of thinking, like, we know that these are happening in tandem. And so they're probably influencing each other somehow. Now, how do I even go about doing this sort of brain body work? Where is the data coming from? This plot is coming from the brain charts paper that came out in 2022. And what they did is they have all these MRIs and they are showing the growth trajectories, or I don't know if they call it growth trajectories, just generally developmental and lifespan trajectories of brain features such as your entire gray matter volume, your white matter volume, subcortical volume, whatnot. And they're just showing you how does this change over time in the same way that we would see like pediatric growth charts for your height and weight. And so the idea is, here is the median for males and females. And so if you are an individual and we know how old you are, we can sort of get a sense of how close you are to the expected brain volume. Now, the only reason I'm showing you this so I can tell you that in 2023, Jenna Shabdak, she was, I think, data analyst in Aaron's lab. So I'm now a postdoc in Aaron's lab. This came out of the same lab a few years before I joined. And what she did is she curated clinical MRIs from the Children's Hospital of Philadelphia. And she went through and looked at all of these MRIs and she filtered out any MRIs that had any signs of pathology. And so she's left with this cohort, which they refer to as SLIP or scans with limited imaging pathology. And she wanted to see in this clinical context, do I get similar features as in my research grade context? And it turns out that they do. They are not finding statistical differences in all sorts of brain MRI features, things like, white matter, gray matter volume, whatnot. The trajectories look comparable, they look the same. And so you can imagine this is a huge untapped kind of resource if you can use clinical data and clinical MRIs, because now, of course, you have all the other data from the rest of the body that exists in the hospital. So let me show you what this looks like. Here are all these growth trajectories. So same thing as with the brain, but for the rest of the body. I am working with about 80 different features that are coming from the body. A lot of these are coming from blood panels or liver panels. We're selecting for panels that are um, asked in routine checkups. So nothing that would only be asked if you were really, really ill, because of course the idea here is to try to make it as general population as possible, with the huge caveat that this is of course a clinical sample, and so we're always going to be like biased towards more clinical populations. So here are some different growth charts for various features. Height, obviously, we know well. Different ones change different degrees. Some don't change at all. Some have this marked change right at puberty. That happens every now and then. We can sort of describe these curves. Almost all of them are monotonically increasing or decreasing. That's the first principal component. This is brain volume. So this is what I will be comparing all of my body features to. And here's the similarity. smaller value means more similar. This is technically the integral of the difference of the derivatives of this curve and all of these curves. And here you can sort of peruse the various body features that I'm using. The number at the beginning refers to the Loink code, which if you don't know what that is, it doesn't matter. You don't need to don't care about it. It's just a helpful way for the hospital to keep track of what feature is being measured. That's essentially what it is. I have been learning a lot about American healthcare systems. This is like, yeah. Anyways, so here are the similarities. And the way that I like to orient myself in this plot is head circumference down here. We would expect the growth trajectory of head circumference to track brain volume quite closely, and it does. It's all the way down here. Where did my mouse go? There. And height is over here. We would also expect, don't forget, this is ages 0 to 20, so only the first 20 years of life. We would also expect height to track brain development relatively closely, and it does also. And this sort of helps me orient myself. Anything below height, I'm like, okay, that's quite a close relationship in terms of how they're growing over time. And then as we go up, that relationship gets weaker and weaker. And this is just over all 20 years, but we can also look at this for every age. Now, this is just a local integral rather than the integral across the entire curve. And the takeaway here is that as we would expect at birth, we are as different in our brain and body as possible. The brain is growing so quickly and the body hasn't quite caught up yet, and we get more similar over time. And then there are these sort of bumps that happen due to like puberty or just differences in our growth trajectories. And the point of this is more of a data presentation here. Here are the features that I'm working with. Here's how they change over time. It's not so much about trying to establish any kind of causal relationships between these, not at all, because I mean, we're just growing, right? So it sort of makes sense that a bunch of features are growing similarly. Now, the main question, though, like what I was more interested in trying to address is whether abnormality is correlated rather than just whether their growth trajectories are correlated. So here is a correlation. I'm correlating how abnormal are you in various body features. I'm doing this across all my 80 or so features. I'm just plotting the statistically significant features. And I'm correlating this with, again, just total brain volume. So if you have a bigger brain than one would expect at your age and sex, do you also have more, let's say, hemoglobin than you would expect for this age and sex? And just so that we know what abnormality means, we have fit these nonlinear models for all of our features. So the brain features for the body features, and abnormality would be like, if this point is you, each point here would be a person, right? If this point is you, you would be, this is a pretty great large deviation from the median. So this would be considered large abnormality. And the question here is, if I have large abnormality in my brain, do I also have large abnormality in my body? Now, big caveat, I'm using the word abnormality. It doesn't necessarily mean unhealthy, right? It just means that you're like deviating from the median. So here are my associations with global brain volume, and we see a lot of blood features coming up, probably related to anemia, like if you're, well, in general, just like less blood, potentially a smaller brain. And then here, what I'm doing is I'm removing the effects of total brain size, which is by far the greatest effect. But once you remove that, then you can start to dig into regional effects. Here, each point here is a brain region, and each column is corresponding to a body feature. And I'm doing this for different regions for cortical volume, surface area, and subcortex. Maybe it's a bit easier to see this on the brain. Here I've only pulled out a few, right? Because every single column here would represent one brain map. But what I'm showing is that there are these effects that are regionally specific between various body features and brain volume outside of the effect of global brain volume. And again, mostly blood features like hemoglobin and erythrocyte distribution width. Now, this analysis doesn't address health at all. So the next question I had was, well, what happens if we pull this into the realm of, does it actually tell me something about whether or not you're healthy? And so what I did there, this is like going to be a little bit difficult to unpack, but here are my body features. What I've done is I've used them to fit models that predict your health. I'm going to call it a risk score. Now, what that really is, and this is sort of what I was alluding to when I said that I've been learning a lot about the healthcare system. In the US, as I'm sure you guys probably know, but I didn't know this coming in. Every time you go to the doctor, you have an encounter and you're billed, your insurance provider is billed. And that billing is given an ICD code, and that ICD code can be mapped to something we call the fee code. And essentially what it does is it tries to tell the system, I suppose, or it just sort of maps your hospital encounter with the reason that you went. So if you went for an asthma-related concern, you would be mapped to a fee code or an ICD code related to asthma. And then those fee codes can be grouped into different broader categories. And these are the broad categories here that I'm looking at. And so what I'm doing when I'm predicting risk, I don't actually know whether these kids are healthy or not, right? Like there's not like a health number. But what there is there's the frequency of these ICD codes. And so if I have somebody who has come to the hospital a lot, like many, many times, and they keep coming for the same concern like asthma, then you can sort of predict that this kid is, has asthma. Whereas if they have only one Phe code hit or one encounter with something and it never shows up again, then it's less likely that they're ill. So that's my outcome. That's what I'm trying to predict. And I'm using these body features, and the logic here is if I know what how the rest of the body is doing, can I predict how ill you are in, or what your risk score is across these different organ systems? This plot is showing how important, so every single dot is the importance of this feature in predicting your risk score related to a given system. So this one over here is musculoskeletal, this one is hematopoietic, color scheme is over here. And then panel A is just trying to tell you that the models are working. There's just a correlation between empirical risk and predicted risk, that's all. So then with this, I can take, my sample of kids who have an MRI, those limited imaging pathology MRIs, I can take those kids and given their lab data or body data in the hospital, which this is not always going to be completely full, right? Like different kids have different amounts of features. I can try to predict their organ risk score. And then once I've predicted that, I correlate organ risk with brain volume. So now instead of correlating just brain and body, I'm correlating brain and how, given your body information, how likely are you to be ill across different systems. And again, predictably, we do see that there's significant relationship between your health, general health, and brain volume. And again, we can remove the effects of total brain volume and look at the regional effects. And here, the main takeaway, for me at any rate, I mean, there are regional effects and we can sort of unpack that. But for me, the main takeaway is that the endocrine metabolic system shows the most associations, the most statistically significant associations compared to all the other systems, which um, makes sense, I think, to me. I sometimes I think about whether I would say that for every single feature, like, oh yes, this makes sense. But at any rate, for now, at any rate, for the endocrine metabolic system, I think it sort of checks out. We would expect there to be large relationships with that in brain volume. So yeah, this is where I'm at. I'm still very much in the process. This is very, very ongoing. Still kind of struggling with the data and trying to figure out where to go or how to establish whether what I'm finding is real and, you know, how much I believe it and do I have to be worried about noise and whatnot. But this is actually my first time talking about the project, so very happy to take any feedback for that. So yeah, I'll bring it back to this slide where I say that multi-scale biological features are measurable and available. And now with my postdoc project, I've sort of taken that to the next extreme, which is to then dig into multi-scale biological features in the rest of the body, given that we have hospital data that has a lot of this information readily available, especially after it was shown that the MRIs are comparable to research-grade MRIs in terms of their growth trajectories. And so my quick little summary here is that at the start, I say that no one person can be an expert in everything. And that is certainly true and something that I still struggle with in the work that I do. But one person can be friends with all the experts. And I sort of say this flippantly, but I do really mean like if you're gonna go down this route and try to do more integrative work, I do strongly recommend collaborating with somebody who does know that data extremely intimately so that they can tell you when you are doing something that makes no sense whatsoever. Because I won't always know. Brad Voytek talks about this kind of stage of neuroscience that we're sitting in right now as being data rich and theory poor. And I agree. And I think that that lends itself to a really nice opportunity to do this sort of integrative neuroscience, to try to bridge all these different scales, to try to bridge these different biological features to better understand the brain from the perspective of an integrated whole rather than these different siloed perspectives. And lastly, that integrating these multi-scale biological phenotypes helps us to answer existing questions like are two regions more likely to be connected if they are expressing similar receptors, but also to ask entirely new questions like what is going on with these synapse phenotypes. For me, at any rate, this is completely new to come in and think about, oh, okay, we have all these different synapse phenotypes. What are they doing? How do the dynamics differ? Does that tell me something about the way I'm anatomically connected to the rest of the brain? And yeah, that brings me to the end. Thanks so much, and happy to take any questions.
Awesome work. Thank you so much. I know I have lots of questions, so maybe I'll kick it off. One of the things that I wonder about in your sort of the sort of combination of the pet data, I know you collected the sex and age, or you have access to the sex and age. Did you look at variability of the coefficient of variation with respect to the ratio of the sexes in your data? Because I know there's some data sets that only have males, some that have half female, half male. There's like different ratios there. So if there
Yeah.
is like a sex difference in the receptor, you might get an overinflated or underinflated coefficient of variation. But I don't know if there are sex differences, period, in these receptor maps as well. So it might be like something to dig deeper into. Because I know that for my
Yeah.
like sex differences work, I would really like to know if there's like, yeah, differences in the receptor maps of males and females.
Yeah, that's a great question. It's funny because when I sent that email, 4 years after the first email and said, hey, like, would you guys be willing to find the coefficient of variation or the standard deviation maps? I remember one co-author sent the standard deviations and then he said, and next you should get the mean and standard deviations for the sexes separately. And I was like, man, like there's just like a whole,
I
this never
know
ends.
it's
Like there's
like
a
never
whole list of
ending, but yeah, I
but
really
it's a
wonder
good
about that. Yeah.
yeah, no, it's a really good point. I think that in the supplement of that paper, I definitely compare it to age. I don't think I compare it to sex difference, but I do know the ratios. You're right. So I could check if the coefficient of variation. I guess I just wouldn't know. I wouldn't know if I were to see a difference or like a correlation there in terms of coefficient, the ratio between inter-individual and regional variability versus sex ratio.
Mhm.
It might be difficult to really pinpoint that it's the sex ratio that's doing it. You know what I
Well, you
mean?
could,
Just given that
I
there's
guess you
so
could
many.
do two separate coefficient of variation maps for males and females and sort of see if they, or
Yeah.
yeah, like do some
If those are,
across.
yeah.
I mean, I don't know, the sample sizes are kind of small for a lot of these. And it's like I said, some of them, I was looking, some of them don't even have females
Yeah.
or they have like
Yeah.
one female. But there are some that have like, 50-50-ish. So you could try to disentangle because you do, yeah, because then it's also confounded by sample size, right? So the variability is going to increase. So you try to
Yeah.
like do some kind of permutation testing with like an N that's fixed or something. I don't know. Anyway, we
Yeah.
can talk about that offline if you want to, but I'm sure there's something to do there. We have a question from William. William says, this is a great talk. I'm just wondering, what are the major diseases or conditions that are represented in the various body systems, particularly for the endocrine disorders? Are the top diseases diabetes and adrenal disorders? Do you know that?
That's a great question. I actually have a hidden slide because
Oh,
I um anticipated this.
oh,
Let's make this
nice.
bigger. Okay, so you were interested in, I mean, this is a little bit janky, but here's the group endocrine metabolic, and here are the different, don't ignore the number. That's just the ICD code essentially that was mapped to the encounter.
Do you mind zooming in? I'm sorry. Just maybe
Yes,
zoom in a
absolutely.
little
Thank
bit.
you for letting me
Yeah,
know. Let
perfect.
me
Yeah, that's good.
there.
Perfect.
And so, oh, I should have added the frequency, but I think yeah, diabetes is here, and I think obesity here. Yeah,
Mhm,
that is another big one.
mhm. Yeah, I saw
Um,
BMI in the top kind of.
mhm.
Yeah. Hmm.
Yeah. Yeah, exactly. So for each one, and this also kind of points to this figure a bit. Like the point here, when I'm showing that the models work, it's more that the bar for each color goes up as your predicted risk goes up. So I just care that things are increasing. The actual count, so empirical risk is going to be like how many ficos you're being hit with. That count is going to vary because for endocrine metabolic, for example, this pink one, that just in general in the hospital system gets hit so much more than everything else. And that's why the value. It's not that the risk is necessarily greater, it's just that there are more fee codes associated with that system, if that makes sense. Let me know if you guys want to see anything else. I can move around.
Yeah, I actually saw that. Oh, William saying, and these are primarily pediatric patients. Yeah, I think they're under 18.
Yeah,
Yeah.
entirely. Yeah.
Could
Well,
you
I
go
go up to
back
20.
up to 20? Okay. Could you go
Yeah.
back to the slide you were just on? Yeah, this guy.
hmm
So I think sex is high for one of those. I can't tell the color. Is it
It's for the
genitourinary?
genital. Yes.
Yeah.
Yeah, exactly.
Okay. Interesting.
And I haven't checked yet, but I should check which sex, like what it's picking up on.
Mm-hmm. Yeah, that's
Yeah, age is high for everybody because I'm using age as like as an offset. Just the older you are, the more
more
hits, the more
visits you're gonna
encounters
have.
you're gonna have,
Yeah.
right? So yeah, that's
Okay.
what that is.
Okay. The other
Yeah,
thing I
this
want, oh, sorry, go ahead.
no,
Yeah.
go ahead, go ahead.
I was just going to ask about there's some work that was recently come out that was talking about the onset of menarche as like a inflection point. So I wonder if you, I mean, is this a linear model?
No,
Uh,
these are all
yeah.
nonlinear.
Okay.
They're coming from like Gamel's model, so it's like a GAM,
Yeah.
but also
Mhm.
modeling the variability.
Yeah, because
You
I just
know,
wonder if there's like a difference in the fits. If you
yeah.
had male and female fit separately, if there would be like a sort of inflection point if you aligned everyone to either puberty or, puberty stage or menarche or something like that.
Yeah, that's a great question. I have looked at I didn't include it here, but I have looked at the curves, these curves for both sexes separately, but for each feature kind of
Mhm.
plotted, yeah, separately instead of together. And for the most part, they look quite similar.
Mhm.
Very obviously testosterone, there's a big difference there. That's kind of jumps out. But just by eye, it wasn't obvious. But
Mhm.
we have been talking in the lab about trying to integrate these sort of different puberty markers. It's just a little trickier because in the hospital system, it's less commonly recorded, like
Mhm.
yeah. So that is the only like I don't know how I would get at that information without doing something like looking at, how
The charts,
testosterone
yeah.
change. Yeah, the charts
Or that,
themselves.
yeah. Okay.
Yeah.
That
But
makes
there's also so much data in the hospital that, I think I know it. And then it's like, oh, there's now this other thing that I didn't take into account. That
I
so
know,
who
I
knows?
can't
Like
even
maybe,
imagine. Yeah.
yeah.
We have another question. This is from Fahima Arab. Thank you. Great work. I'm wondering if you have considered using causal graphical models like Bayesian networks or structural causal models to disentangle relationships among brain and body features instead of relying on pairwise correlations for their predictive power. Yeah, it's a question I had too.
That's a great question. I haven't. I would be curious to tell me more. Like how would you approach that?
I can unmute Fahima if you want to, but I think it's more like, yeah, I'm thinking of like a mediation analysis or,
Hmm.
yeah, some kind of structural equation model, whether you do like a kind of
Right.
set order of linear regressions to try and disentangle. I mean, it's tough too, but you do have this longitudinal data that I'm sure you can kind of potentially leverage to get out causality, I wonder. Because I think you're looking
Maybe.
cross-sectionally right now, if I understand correctly, or
Yes, it is mostly cross-sectional, but it's true that there are some individuals that have multiple scans. And
Mhm.
so what I'm doing is I have a scan date and then I'm looking around like a year of that date for any lab measurements because it's not necessarily the case that they got the like blood
Right.
test at the same time they got their scan. And so it is possible for probably a relatively small but still good enough sample where I have multiple scans and potentially also multiple kind of lab
Hmm.
measurements.
Hmm.
Yeah, I could definitely look into that. It would be really interesting, and it seems like a shame not to, given that it exists.
Such a rich data set, yeah.
Yeah.
All right, we have another question. Great talk, Justine. I know you did not want to mention environmental factors that might influence the data you've collected, but is there anything you might be working on that you can discuss?
Yeah. No, I'm more than happy to talk about environment. So one thing I haven't done but need to do is just generally looking at effects of social economic status. And one way to get at that is that we have for these patients geocodes. So we kind of have a sense of where they live approximately. And as from there, you can get a sense of, neighborhood green space and I don't know, schools in the area, walkability, things like that. And try to get kind of an SES metric there. And I'm sure that there are strong effects, especially given that this is the Children's Hospital of Philadelphia, and Philadelphia has a really wide range of socioeconomic backgrounds. So that I think that is as far as I will go down that road, just like assessing what are the relationships, what relationships, can we disentangle SES from the brain body associations, or maybe we can't, in which case it's just sort of, we don't know. Um, and then after that, I don't know yet if I will go down the road of actually trying to make associations with the environment that go above and beyond SES. It just gets really muddy once we go down, what is SES and what's not, and that is what makes it a lot trickier. But I definitely want to know. I'm a little bit nervous, I'm not gonna lie, to find out, but I want to know like what the associations are.
I think that's well said. We have another question from Tracy Butler, sort of a side question. I have plasma PSD 95 measures, and I'm trying to understand what this might mean in terms of inter-individual differences in regional brain volumes and metabolism. Any knowledge or opinions about this?
Oh, okay. Wait. You have this per person or spatially across brain
I
regions?
think plasma, so it's a blood
Oh,
marker,
it's
single
right. So
measure.
it's per
Yeah.
person.
Hmm.
Oh my goodness. I don't know. I'm sorry. Plasma PSD95 across people. Yeah, I suppose. Well, I guess when I like struggle with individual variability, one of the things I'm always thinking about is like how much does it vary and how much should it vary, and then how much do I care that it varies, you know? Because there's an element of like there's going to be variability, but what's your like biological question? What parts you actually care about? And in some cases, I think we don't necessarily care so like we care more about the similarities than we do the differences. But that's just generally individual differences, not so much specific to plasma 95.
She
Yeah.
she followed up with, okay, I don't know either.
Right.
Good, yeah. TBD. And then Fahima was coming back to about the mediation, the causal sort of. There are multiple methods in causal discovery that you can use for cross-sectional data as well.
Yeah,
And then
I
we
should
have,
look into that.
yeah, you should, yeah, that'd be really interesting to see. There's another question from Joanna. Thank you for the fantastic presentation. Are you interested at all in comorbidity of mental disorders versus physical diseases, maybe with correlation to your current data?
Yes, definitely. Yeah, one of the challenges with this SLIP, so this limited imaging pathology, scans with limited imaging pathology cohort, is that because they're all collected at a hospital, it's probably really biased towards things like mental and neurological, but neurological has been a little bit corrected for because we've removed scans without obvious pathology. So a lot of these could be more related to mental disorders or trying to diagnose mental disorders. So it does sort of lend itself a nice opportunity to dig into those questions, but also I know there's always like two sides of the coin, right? On the one hand, cool questions can be asked. On the other hand, I need to be a little bit careful about any conclusions that I draw because my sample is likely biased in that way. I have been thinking about, I played around with a very epidemiological question. It had nothing to do with the scans, but I was just looking at which fee codes, so which kind of, again, that's like the thing that comes from the ICD code, which comes from your encounter, your hospital encounter, and tries to label like why you came to the hospital. I was trying to look at co-occurrences of those. Like if you are getting a lot of one, what are you also getting a lot of in like for another thing, an age and sex kind of stratified. And there was definitely signal there. There are definitely, certain anxiety disorders and certain other mental conditions where I see across organ systems different frequencies, like greater or less than you'd expect by chance, that are coming, this like comorbidity idea. I don't know if I'm going to do anything with that yet because right now it's just sort of sitting in a PowerPoint as a figure of something I just did for fun and had nothing to do with the scans. So that I never got back to it, but it's on my mind. It's kind of simmering back there, and then we'll see if it reemerges. Yeah.
I think that would be super interesting to see.
Yeah.
Maybe, yeah, in the SI, right? So he's a, no,
Yeah, I know,
maybe,
right?
you never know if it's an SI figure or a full paper. Yeah. We have another question from Melissa Walsh. Thinking about the brain as an embodied organ rather than an isolated system, have you had the opportunity to analyze any whole body PET data sets in your work?
Oh my gosh, I haven't. I've just attended talks where people talk about it and my mind is blown. That's like the extent to which yeah. Even MRI, like with this heart thing, right?
Yeah.
Which is like, oh, let's just segment the heart and see how that is really. That just complete, yeah, it blows my mind, but I haven't yet. That would be really cool.
Yeah. Very good. Okay. Good. Okay. I don't have any more questions if people
I have
have. Oh,
one
yeah,
more
go ahead.
question, Justin. Great talk. It's inspiring all the things that you're doing. I'm wondering if you are thinking of doing a similar work with older patients. I guess you are working with pediatric patients because you are interested in how, like, the brain development is co-evolving with other systems, but do you have access to other patients? And yeah, I work
Yeah.
with like pregnancy and parents, so I don't know, curious if you start working with other patients, you can also know if if they are parents or not parents, because that's like a huge health inflection point. So yeah, just
Absolutely.
curious.
No, that's a great question. Well, actually, it's funny that you say that you mentioned pregnancy because this is a pediatric hospital, but pregnancy we do have because a lot of mothers come to
Mm.
obviously give birth to their children in the hospital, and so we have, a bunch of kids and then a bunch of pregnant individuals. I haven't looked at it yet, and I think the only way to extend the analysis to the whole lifespan, which I think would be really interesting, would be to do something like to go into the UK Biobank. I don't know how I would do that with a hospital context. Because one of the only reasons I can use this hospital data is that because I'm at CHOP. And so I just don't know like what other hospital would, let me sort of dig in their archives. Um, but to be fully honest, when I came to the project, I wasn't approaching it from a developmental perspective. I was approaching it more from just like a curiosity-driven brain-body association. So the fact that it was over a development wasn't by design. It was more so that that is the data that was there. And then I realized after the fact that that's actually very interesting because so much is changing. And so then you get these really interesting associations that show up. But I would absolutely be interested to see how it looks over the rest of the lifespan. It just would be difficult, is my like fear, I suppose.
Of
It would be a
course.
whole other
Yeah.
thing. Yeah.
So you don't have access to that data through your hospital.
The hospital doesn't have people who are much, at any rate, the sample sizes for anyone who isn't pediatric drops off very, very quickly. So the hospital doesn't have the data,
Mm,
but I mean, the UK Biobank does, and I think that I've never
yeah,
actually worked in the UK Biobank, but I
yeah.
think others in the lab have.
But
So,
the UK Biobank starts like I think the
right.
younger it's like 40
Hmm.
something. So yeah, there's
I would
always
have
this
20
gap
years.
between like 20 and 40 something, which are the
Yeah.
reproductive years. But that's just the way
Yeah,
data
yeah.
is. Yeah,
I wonder
but
if I
yeah,
could dig into I don't know how easy or how accessible it is to find the all the hospital encounters for the pregnant individuals, but that could be really cool. I haven't tried. Yeah.
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 Okay. If there aren't any other questions from the panel, there's one more question from Susan Gordon. Are you interested in changes in brain chemistry during reproductive transitions in women across the lifespan, which is kind of what we just talked about, but
Yeah.
yeah.
Yeah, I mean, with any kind of big change, these
Mhm.
are like the most sort of exciting periods of time to be looking at, right? And like one of the things it hasn't quite panned out yet, but one of my questions coming into the project before I started it with was if I find, brain and body features that are associated over time, how do those associations change over time? Do they emerge later? Are they there at birth and then they disappear? Do they come in with puberty? Do they leave with, whatever? Um, and unfortunately, I come from like the brain imaging world where I was just correlating brain maps and all the correlations are like zero point 0.6, and then now I'm correlating across people and like if you get zero point 0.2, you're like really excited. And obviously, I need to like that's fine and normal, and like you wouldn't expect a zero point 0.6 correlation across people because that would be a huge amount of variance to be accounting for, and that would be kind of crazy. But it takes a little bit of my own like readjustment of what I believe in. And so one of the things I've been struggling with a little bit is correlating things across people, getting really small effect sizes even if it's statistically significant. And then I'm like, well, if it's such a small effect size, do I want to call that coupled? And do I want to interpret a decoupling? You know what I mean? If it's not like zero point 0.9 correlated to 0 correlated, it's like zero point 0.2 to 0.18 or whatever. Um, so I'm still sort of struggling with that and like readjusting my expectations of what the magnitude of a correlation should be. But definitely am interested.
Good. Okay. Then I think if we don't have any other questions, we can stop there. And thank you so much again for giving that wonderful talk. It was really interesting to see. And have a good week, everyone.
Thank
Thank
you
you.
so much for inviting
Bye.
me.
Bye bye.
Bye.